Welcome to part 5 of the disparate correlation training. MIADE, minimum information about disorder experiments. Here you can find the learning objectives of this part of the training. Discuss what MIADE is and its purpose. Define and give examples of sequence construct information, experimental conditions definition, experimental components definitions, and interpret MIADE examples in disparate. Let's learn more about MIADE now. MIADE is minimum information about disorder experiment. Was developed by the Elixir IDP community. You can find more about the community and its goals and activities here. It's guidelines for data producers, biocurators, and database developers. And it was developed, created to enhance accuracy and accessibility of intrinsically disordered regions annotations. So the aim of MIADE is to ensure the most accurate annotations with all the details that are relevant for each annotation. We recommend to refer to the MIADE guidelines for more detailed information about the guideline itself, that you can find also linked here. So let's start with sequence construct information. It's used to define, annotate differences in protein sequences defined by MIADE. And from the correlation interface, curators will be able to select factors from the drop-down menu, the corresponding sequence construct drop-down menu, as tag labels, mutations, PTMs, non-standard amino acids. So you can find all these alterations and you can specify them. For example, you can specify which type of mutation, let's say a substitution, a deletion, an insertion, and also you will be able to specify the specific mutation that you found in a publication. An example is the cellular tumor antigen P53 linked here, that is protein entry. One of the annotations in this protein entry shows also with MIADE the annotation of 4 non-synonymous substitutions in the construct. Then we have experimental conditions. Experimental conditions are again specified using a drop-down menu, and they can include pH, temperature, redox potential. So, considering these conditions, it's also possible to add the absolute values and deviations. And again, there is a drop-down menu selection for parameters. An example of this protein entry linked here is the hemagglutinin protein that is reported at pH 5 as an experimental condition. Finally, we have experimental components. So far, we have 7 sample components defined by MIADE that are specified again using drop-down menus in the curation interface in disparate. You can use the drop-down menu for selection of components. You can add also concentration and deviation if relevant. And you can select, for example, an interacting protein partner, an interacting small molecule. For example, here we have again the example of P53 from disparate, and we have an interacting small molecule and the identifier from the resource for this specific interactor. Let's see now some examples of MIADE in disparate. These are entries, pieces of evidence that are available in the resource, so you can also search for them. Here you see an example of structural state annotation. This is the disordered region. This is the experimental technique annotated with MIADE. This is the term disorder. The region is disordered. A cross-reference from PDB. And it's indeed the hemagglutinin protein that we just mentioned in one of the previous slides. You see that the fragment is studied at pH 5. And this information is available here. And also statements from the publication that define the intrinsically disordered region as well as the pH that was used in the experiments. Again, here we have another annotation for hemagglutinin. This time you will see that the fragment is the same, the region is the same. The technique used to study the region is always X-ray crystallography, but we are describing an ordered fragment now, an ordered region, because the pH now it's 7.5, and at this pH the fragment is not disordered anymore. So here you can see also a transition. There is no MIADE information necessary here, but this annotation describes the transition between the two annotations, previous annotations. So order to disorder. We are defining a transition from order to disorder. This is the region, the fragment, with this technique, X-ray crystallography. These are the statements from the publication that support our annotation. And this is a transition between the source evidence, which is the ordered piece of evidence, and the target evidence, which is the disordered one. Again, here, from another protein, we have a disordered to ordered transition from this region that was studied with Fred technique. And you can see several interesting annotations with MIADE here, because we can see 3 different types of construct alterations and an experimental component. So for what concerns the construct alterations, we have a tag. We know that it's a his tag in a C-terminal position. We have a protein mutation, which is a substitution of alanine at residue 389 in cysteine. And then we have labels and dyes, fluorescent dye label position 389. Then we have an experimental component, which is the interacting protein calmodulin 1 and the corresponding uniprot accession number of calmodulin 1. Here again, we have a molecular function, so an annotation with a gene ontology term. The term is calmodulin binding for region available here. The type of evidence here was mutant phenotype evidence used in manual assertion. And you can see that here we have indeed several mutants. So every arginine that was changed in alanine, but we also have the experimental component interacting small molecule calcium.